Hello, and a very warm welcome to everyone joining us for today's Select Science webinar titled How to Revolutionize Cell Therapy Analysis with the Power of Double-Emulsion Flow Cytometry. My name is Jemima Arnold, and I'll be moderating today's presentation. I'm delighted to be joined by our guest speakers, Dr. Bárbara Schlicht from Samplix and Dr. Garret Guenther from Agilent Technologies. This webinar will discuss how Xdrop and NovoCyte enable cytokine secretion assays as well as cell-cell interaction assays. Furthermore, our speakers today are going to explore how this innovation has the potential to transform single-cell droplet microfluidics by expanding the scope of single-cell functional screening assays and unlocking new frontiers in the understanding of single-cell behavior and function. Following the presentation, we will have time for a question and answer session. So if you have any questions throughout the webinar, please feel free to submit these in the box to the right of your screen. Without further delay, I would like to hand over to our speakers, and I would like to thank them again for presenting for us today.
Hi, welcome everybody. My name is Garret Guenther with the Agilent flow cytometry team. And before we jump into double emulsion flow cytometry, I just want to introduce you to the Agilent flow cytometer solution used to generate some of this interesting data. And quickly, I want to mention that we have an application note highlighting some of the double emulsion flow cytometry data that will be presented today. And this is available on agilent.com.
Agilent NovoCyte flow cytometers have been around for almost 10 years now. They have different levels of capability, coming in 1 to 5 different laser options and up to 30 fluorescence channels. One thing to note that the NovoCyte Quanteon, Quanteon, and Penteon systems listed below here incorporate advanced detector technology called the silicon photomultiplier to detect fluorescence, and they are the only flow cytometers out on the market that have been able to utilize these state-of-the-art detectors. These NovoCyte systems are known for their sensitivity, fluidic stability, absolute counting, fast electronics, and the many automated functions, including routine maintenance. NovoCyte systems are also known for being easy to use. Experimental setup is easy by not having to adjust detector settings so samples are on scale. The software is easy to use to navigate both sample acquisition and data analysis. Routine maintenance is easy with the push of a button, and sample acquisition is easy with the help of a versatile autosampler, allowing for true walk-away capabilities while the NovoCyte does the work for you. To make flow cytometry experiments even easier is a new feature we've recently added called EasyPanel. This is a tool freely available on agilent.com that can make complicated panel building even easier. To do this, first you select your instrument configuration. Then add the markers included in your panel. You can customize the fluorochrome, antigen density, dump channels, and marker grouping, or just let EasyPanel make the determinations. For example, let EasyPanel determine the antigen density based on published literature. The next step is to define if you want to use a viability dye or not, and whether you want EasyPanel to decide which one. Then let EasyPanel determine the most optimized panel, taking into account pairing bright fluorochromes with low antigen density and vice versa, fluorescence spillover, and any groupings you have defined in the setup. A panel is provided to you with visible spillover spreading matrix, as well as the score so you can compare between different panel options. We at Agilent have designed the NovoCyte flow cytometers around making flow cytometry easy without compromising on data quality. Now with that, I would like to hand it over to Bárbara so she can show you the power of double emulsion flow cytometry.
Thank you, Garret. Hello, everyone. I'm Bárbara Schlicht, a product manager at Samplix. Today, I'm going to give you an introduction of Samplix technology and how the combination of droplets together with flow cytometry can open new frontiers in different research applications. Let me first introduce you to Samplix. We are a multidisciplinary team of experts focusing on the development of tools for biological research. Our expertise is in both droplet microfluidics and cell biology. We are currently based in Copenhagen, Denmark, as well as California in the US. We are all here to tackle a critical challenge: how to decrease time from research to therapies. Whether it's about testing viral vectors, validating insertion sites, analyzing cell-cell interactions, or selecting your best natural killers, the role of single-cell technology is pivotal in precisely isolating live cells based on their function. By seamlessly integrating high-throughput single-cell screening into early research, the development of gene and cell therapies gain an informed and efficient edge, significantly accelerating the entire process. At Samplix, we strongly believe in this integration, enabling a swifter journey towards breakthroughs. Our mission is to empower research towards cures by facilitating the function-first screening of millions of variants per day through our revolutionary Xdrop technology. Today's agenda will revolve around 6 main topics: how droplet microfluidics enable high-throughput analysis, an in-depth look at Samplix product offerings with the Xdrop family, exploring the potential of double emulsions in flow cytometry, and 3 case studies. One describing single-cell cytokine detection in droplets, followed by a cell-cell interaction assay, which is described on the app note with Agilent, and finally, describing how assays can be multiplexed in droplets. So let's start with droplet microfluidics. Droplet microfluidics is an integral part of what we do here at Samplix. Many areas of modern research rely on high-throughput analysis, meaning that thousands or even millions of individual assays need to be performed in order to screen samples and increase analysis depth. Performing high-throughput assays using tubes or microtiter plates, which allows multiplexing, can take months or days and use up high volumes of reagents. With all these factors added up, the assay also becomes very costly and labor-intensive. Droplet microfluidics allows substantial assay miniaturization and parallelization. Assays in single cells can be compartmentalized in thousands of picoliter volume droplets per second. This increases assay depth, allowing researchers to explore a larger experimental landscape, all while decreasing the volume of reagents used and waste produced. Samplix focuses on the generation of double-emulsion droplets. The process of double-emulsion generation involves 2 emulsification steps, beginning with the formation of a primary water-in-oil emulsion, where the aqueous phase, here shown in light blue, contains your samples or cells. This is then further emulsified in a second layer of aqueous buffer, in darker blue. The precise geometry of our cartridges and pressure controls ensure the generation of thousands of monodispersed double-emulsion droplets per second. Due to its monodispersity, these droplets can then act as individual vessels or microreactors, like individual wells on a microtiter plate, where single cells can be encapsulated, cultured, and analyzed. Moreover, particularly for double emulsions, since they are suspended in an aqueous outer phase, these droplets are then compatible with flow cytometry and cell sorters. Now let's talk about the Xdrop family of products. Samplix offers 2 platforms for droplet-based workflows. The Xdrop, which is a droplet generator used for sample encapsulation, for example, for the encapsulation of single cells, and the Xdrop sort, which is a droplet generator and sorter for high-throughput screening applications. The Xdrop sort is capable of sorting droplets based on a fluorescent output. Positive hits can then be analyzed further by sequencing or culturing. Droplet generation and sorting requires the use of appropriate cartridges and reagents, all available from Samplix. Our cartridges enable 8 different droplet productions or cell encapsulations at the same time, allowing sample parallelization and increased throughput. We also have digital tools available on our website which aid the user in preparing and analyzing their assays. Here's a short video demonstrating cartridge preparation for droplet production at real time. For sample preparation, you require Samplix cartridge and Samplix reagents. Each cartridge can run up to 8 different samples at the same time, so you can prepare it for either just one sample or all 8. Only 3 reagents are required for cartridge prep. You can either use a regular pipette or a multi-channel pipette if you want to speed up the process. Once the reagents are in, a gasket is fitted on top of the cartridge and it can be placed in the instrument. The user selects the desired programs and the cartridge lanes to be run. After a few minutes, your droplets will be ready for further processing. So now let's talk about droplet flow cytometry. As mentioned earlier, double-emulsion droplets are dispersed in an aqueous atom phase, which allows integration with flow cytometry. The advantage of using droplets for cellular workflows in flow cytometry is the ability to analyze the cellular environment whilst retaining the genotype and phenotype connection through the whole workflow, even when the molecule of interest is not directly bound to the surface of the cell. This compatibility significantly enhances the capability of flow cytometry, enabling analysis of the cellular environment, quantification of secreted protein, as well as cell-to-cell interaction assays. Allowing the analysis and sorting of droplets using conventional techniques ensures that researchers do not have to make drastic changes to already established protocols. This compatibility highlights the user-friendly nature of the technology, making it more accessible and appealing to researchers across various fields of studies. The encapsulation of sample or cells and their microenvironment within these droplets significantly expands the potential assay types feasible for analysis. The utilization of double-emulsion droplets amplifies the capability of flow cytometers by facilitating the study of cellular environments, quantification of secreted cells, and analysis of cell-cell interaction assays. Given the widespread usage of flow cytometry techniques in research settings, offering this opportunity ensures that users can integrate Xdrop platform in their workflow without the need for extensive protocol changes and still take advantage of multi-channel flow cytometry analysis. Most of the assays that I will describe here have been analyzed using Agilent's NovoCyte in a very simple workflow. First, sample and reagents were pipetted into the Xdrop cartridge, placed in the instrument for encapsulation, incubated, and then analyzed by flow cytometry. So let's talk about some applications, starting with cytokine secretion assays. This assay was originally developed for a bulk cell assay, and using the Xdrop, we have turned it into a single cell assay. So single cells were encapsulated using kappa antibodies bound directly to the cell surface, together with cytokine-specific detection antibodies in droplets. These droplets were then incubated, and the cytokines secreted by the cells were detected by a fluorescent signal as it bound to the detection antibodies. Performing this assay in droplets allows for single-cell resolutions, as cytokines cannot partition from one droplet to another. Therefore, we can be certain that all the cytokine detected has been secreted by that single cell. After incubation, droplets were broken and its contents released. Any unbound antibody was washed out, and the levels of cytokine production by each individual cell was analyzed by flow cytometry. In this particular experiment, we have taken T cells from PBMCs and stimulated them to secrete TNF-alpha. After flow cytometry analysis, a population of highly potent TNF-alpha secretors could be clearly identified from the average population. When the same experiment is carried out in bulk, only an average increase could be noted. By doing single-cell assays, true population heterogeneity can be identified. In this specific example, it can be concluded that the majority of the cell population are not potent TNF-alpha secretors, and therefore, the majority of the cell killing is done by a small part of the population. This assay can also be multiplexed by the addition of multiple detection antibodies for different cytokines. I encourage you to visit Samplix.com and read our app notes on single-cell multiplexed cytokine assays in droplets. Now that we have identified our high secretors, we want to see if these high secretors retain their potent profile after expansion. So we have sorted out the high secretors, expanded them in culture for 2 weeks, and after the 2 weeks of expansion, they were stimulated again and the cells were tested. The sorted high secretors have kept their potent profile even after 2 weeks of expansion. Now let's talk about cell-cell interaction assays. This assay now features on the joint app note co-authored between Samplix and Agilent, shown by Garret earlier. This assay requires a co-encapsulation of 2 different cells in one droplet to analyze how they interact with each other. Here, we want to identify our best killers. In this case, we have taken an NK cell, stained in green, and co-encapsulated them with a K562 target cell, stained in red. They were incubated in droplets, and killing was measured over time by the detection of propadian iodide signal, indicating cell death. These microscope images show the same droplet containing both cell types. We can identify each cell by their different staining and detect the death of the target cell by the propadian iodide signal. This assay was analyzed by flow cytometry. 4 different droplet populations were identified. A population containing only empty droplets. Next to it, in the high green signal, we can identify the population with only NK cells. In the high red signal, we can locate droplets containing only target cells. And in the high green and high red, we can identify droplets containing both cell types. When we take a closer look at the droplets containing both cell types and the PI intensity over time, it can be seen not only that the IL-15 activation increases the cell killing ability, but also a population heterogeneity can be identified based on the speed of killing. 4% of the population were able to kill the target in the first hour of incubation. 14% were medium killers, and 25% of the population were slow killers, only managed to kill the target cell after 24 hours of incubation. We can also look at the droplet population containing only one of each cell type, either one NK cell or one K562 cell, as in-built assay controls for cell survival in droplets. We monitored these individual populations and analyzed cell death for the same time points as the co-encapsulated populations. We can see that for NK cell, there's only a slight increase in total cell death after 24 hours of incubation. And for the K562 population, no significant cell death was noted for the first 4 hours of incubation, and only after 24 hours, around 7% death was noted. The final assay I'm going to show you is a granzyme B assay. Granzyme B is a potent cytotoxic enzyme secreted mainly by T lymphocytes and NK cells. They are secreted alongside with pore-forming protein perforin to mediate apoptosis in cancerous cells. For this assay, we have used a similar workflow as the previous killing assay, but instead of detecting cell death, we detected granzyme B secretion. So a target cell and an effector cell were co-encapsulated in droplets together with a granzyme B substrate. Granzyme B secretion will then be detected by the cleaving of the substrate, which releases a fluorophore that can be detected by flow cytometry or fluorescence microscopy. From the fluorescence microscopy images, it can be seen that granzyme B positive and negative droplets can easily be distinguished. Within the positive population, different levels of fluorescence distinguishes different levels of NK activation. If we take a closer look at a droplet containing a high granzyme B signal, we can see that they contain an NK cell and K562 cell debris shown in red. This assay can be multiplexed by combining with the previous cell killing assay. If we take a look at the co-encapsulated population again, where both cell types are contained within one droplet, we can appreciate different populations within this gate. A population containing no cell death and no granzyme B, a population containing cell death indicated by a high PI signal but no granzyme B, a population containing cell death and granzyme B signal, and another containing only granzyme B signal. When cells have been stimulated with IL-15, we can see an increase in granzyme B secretion after 30 minutes. This increase leads to a sequential increase in cell death after 4 hours. No increase in cell death is seen without any granzyme B after 4 hours. In summary, we have demonstrated how droplet microfluidics can be used for single-cell assays. In particular, how Xdrop, together with flow cytometry, can be a powerful tool for functional analysis of single cells, enabling a large range of assays to be performed, including cytokine secretion assays, cell-killing assays, granzyme B secretion, and combined multiplexed assays. It is important to reiterate that these droplets can be sorted, and after sorting, cells can be released from the droplets and expanded for further analysis. To learn more about our instruments for droplet generation, please visit our website at samplix.com. Thank you for your attention. If you have any questions for either Garret or I, please enter them into the chat and I'll be happy to answer.
Thank you for that very informative presentation, Bárbara and Garret. It is now time for our question and answer session. So if you have any further questions, please do send those in. You can submit these by typing into the questions box to the right of your screen. So the first question I have for you guys is, what are the advantages of analyzing cells in droplets?
Hello, I think I can answer that. So it's Bárbara here. So the main advantage of analyzing cells in droplets is that the full cell secretome, or whatever the cell secretes, can stay contained. So you can analyze the cell as well as all the proteins it secretes as well. You can do also quantification analysis of this protein, so see how much of each protein the cell analyzes. As I showed in the presentation, another thing you can do is cell-cell interaction assays. So you can see how two cells interact with each other. Thank you.
Useful, thank you, Bárbara. The next question I have for you both is, can cells be taken out of droplets?
Yes, so at Samplix, we sell some reagent kits, and one of the reagent kits we sell is a droplet break kit. And what this kit does, it breaks these double-emulsion droplets, releasing the cells so that it can be the cells can be seeded and expanded as the customer wishes. Or you can do any transcriptomics afterwards with the cells, or genomic analysis.
Thank you for explaining that one for us. I have another one here with somebody asking, how many different samples can you prepare in each Xdrop cartridge?
So the Xdrop cartridge is designed to prepare 8 different samples in parallel. So you essentially can run 8 different experiments in one cartridge. One cartridge takes about 8 minutes to run. So within 8 minutes, you have 8 different samples that you can then experiment and analyze.
Great, thank you, Bárbara. And then I think our final question for the day is, how big is the Xdrop instrument?
The instrument is a small instrument. It's around 30 centimeters tall and not much longer. So I think it's about 40 centimeters long. It fits in an LAF cupboard, for example, or in a bench, like laminar flow bench. That's what I mean. So you can keep all your experiments clean and sterile by positioning the instrument inside of one of these hoods.
Beautiful. We've just had quite a few more questions come through, so I feel like we'll just carry on as we are. So the next one I have for you is, what is the maximum number of cells that are processed per sample at a time on the droplet generator?
the maximum number of cells that can be processed at a time. So I don't know if we have a maximum for that. It depends. So usually people want 1 cell per droplet, and we produce about 1 million droplets. So I would say somewhere around 1 million droplets, depending how many cells you want to analyze or to encapsulate in the droplets.
Lovely, thank you. And then another one asking what the double/triple rate is, and what and does that depend on the size of the cells?
So our cell encapsulation in droplet follows a Poisson distribution. Poisson distribution is basically it's a normal distribution. So by determining the initial concentration of your cells will determine what percentage of droplets that you have, which are empty or which contain 1 cell or which contain 2 cells. So it will depend on the initial concentration of your cell sample. You can dilute it enough to make sure that you have mostly empty droplets and droplets with 1 cell only. So you minimize the number of doublets or triplets in your droplet. It follows Poisson distribution, and we have a calculator, a cell calculator in our website, which helps the customer define the initial concentration of cells that they need to use.
Thank you, Barbara. Then I have another one here asking, does droplet generation increase the sensitivity of flow cytometry?
I guess it doesn't increase sensitivity, but allows you to explore a different part of your experiment. So because you'll be able to then analyze the cellular environment, it means then you can do that using flow cytometry. So you can analyze the full droplet in flow cytometers. It does increase the assay time because it's a small volume. It means that you have the time to assay is decreased, so you have a signal much earlier than if you did in a microwell plate where the volume is much larger. So when we talk about droplets, we talk about picoliters, and of course, microtiter plates is microliters. So, yeah.
Thank you. And then another one here asking whether this technology is only good for functional assays or whether it provides any benefits for simple surface staining characterization of cell markers.
You can use for a single cell surface staining characterization as well. So we focus on functional assays just because this is what our expertise lie on, and what we try to bring app notes about, but you can use for other applications as well.
Thank you. And then another one asking, what size double emulsion droplets does it make?
We have 2 cartridges. One that we call a D20 that makes small double emulsion, so they are about 1.6 picoliters, and then we have the D50 that makes droplets of around 100 picoliters. So the larger one is better for mammalian cells or larger cell types. The smaller ones we usually suggest for people working with genomic workflows or microbial cells.
Lovely, thank you. And then a final question here asking whether this method can be used for exosomal studies also.
I'm unsure what this question means, but you can feel free to contact us and I can try to expand more on this question. I'm sorry, I cannot answer that right now.
No worries, that's all we have time for today. So I'd like to thank our expert speakers for the informative presentation and discussion, and a big thank you to everyone joining us online. We hope you have found this a worthwhile session. If you have any further questions, please feel free to email me at editor@selectscience.net and I'll follow up with your questions for our speaker. Remember, you can download related resources in the tab to the left of your screen, and this includes the certificate of attendance. If you'd like to listen again to today's webinar or invite a friend to listen, it will be available to watch on demand in just a few days. Goodbye, and thank you once again for joining us.